makes up a significant part of our lives. Not only is it important for our survival, but we also enjoy eating it. Here we see some foods that are grouped together based on their sources of organic molecules. These foods here are a good source of protein. These foods are a source of carbohydrates. And these are a source of fats. Let's take a look at these organic molecules and what they mean for us. Organic molecules are made and used by living things. For our purposes, we'll look at 4 organic molecules: carbohydrates, lipids, proteins, nucleic acids. As we do, what we'll try to see is their structure, like what makes up your organic molecule. Are there categories that we can, more than one kind or type of that organic molecule? And then how does our body use it? Like what's the use in the human body? So let's take a look at carbohydrates first. What kind of structure does it have? Well, notice that it's made up of carbon, hydrogen, and oxygen. We've got a ring-like structure for the carbons, and there's going to be a particular ratio of carbon to hydrogen to oxygen. In this case, for every carbon to hydrogen to oxygen, it's a 1 to 2 to 1 ratio for carbohydrates. There are several different types of carbohydrates. The simple sugars, or monosaccharides, mono just means one. The disaccharides, which means 2, a double sugar. And the polysaccharides, which means lots and lots of those glucose units stuck together, or the simple sugar units stuck together in long chains. Examples of monosaccharides are glucose, fructose, and galactose. Examples of disaccharides include things like sucrose, what you would find in table sugar, lactose, a sugar you'd find in milk, or maltose, grain sugar. Polysaccharides include things like cellulose, which you would find in leaf products and plant products. We can't digest cellulose. We don't have the enzymes or the capabilities to get energy from cellulose. So why do we eat things like lettuce or apples, besides the fact that they taste good? Why do people say an apple a day keeps the doctor away? Because we don't digest that fiber or that cellulose, but it does help to clean out our colon and keep us having regular bowel movements. Starch is the carbohydrates that you'd find in things like potatoes and spaghetti. We get lots of energy from that. And the energy that's stored in our liver and often our muscle cells is glycogen, this long, complex polysaccharide. What do we use carbohydrates for? Why do you want to eat them? Well, the first reason is because they're the main source of energy in our body. They give us 4 calories for every gram. So for every gram of carbohydrates that you eat, you'll get 4 calories of energy from that. And about 60% of your diet should be carbohydrates. So you'll have plenty of energy to do all the body functions that you need to do.
First category is carbohydrates. We use these typically for energy. They can be divided into 3 major groups: monosaccharides or simple sugars, disaccharides or double sugars, and complex carbohydrates. To start the Benedict's test, we'll first fill our test tubes with some of our simple solutions. We're going to put water in test tube 1, cornstarch in test tube 2, table sugar, sucrose, in test tube 3, honey, glucose, in test tube 4, gelatin powder in test tube 5, and egg white into test tube 6. After adding the Benedict solution and heating the test tubes for 2 minutes, we see the results. We're looking for a red, orange, or yellow color. That's a positive test for simple reducing sugars. Honey has a positive test because it's a simple sugar. Adding iodine is an easy way to tell if there are starch present. Starches will turn black in the presence of iodine. Here we see that the cornstarch turned black and the saltine cracker turned black because both have complex carbohydrates like starch present. You're looking at a slide of starch granules in potato cells. Starch is just a common way to store products like glucose. Plants store glucose, and we get that energy from eating things like pasta, bread, even potatoes.
Take a look at lipids then. The structure of lipids is different than carbohydrates. Still made up of carbon, hydrogen, and oxygen, but the structure is different. Here we have long chains of carbon, and the hydrogens all around them in a ratio of 2 to 1. If all the electrons on the carbon atoms are bound to hydrogen, we call those saturated fats. If sometimes there's a double bond between the carbon and the hydrogen, we call that an unsaturated fat. In other words, we could add more hydrogen to it. There's another chemical bond available. Saturated fats are typically animal products. They're solid at room temperature and generally bad for you. Unsaturated fats are typically plant products, liquid at room temperature, and good for you. What we've done is we've taken unsaturated fats, we've added some hydrogen to them, and to make them a semi-solid, and those are called hydrogenated oils, or things like margarine. How do we use fats in the body? You might think, oh, I don't want any fat in my diet. No, 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 that's bad. But your body does need and use fat for things like cell membrane. You might remember the lipid bilayer of the cell membrane. Those lipids are required for that membrane to have any kind of integrity. Steroid hormones, which sometimes elite athletes, not having a high enough body fat percentage, especially female ones, will be unable to make some of their estrogen hormones because there's not enough fat available. And we need those hormones. Insulation, to keep you nice and warm, we get 9.3 calories per gram from lipids. And about 25% of your daily calories should come from good sources of fat. So there's unsaturated fats as opposed to saturated.
We typically think of fat. This is what we think of: saturated and unhealthy fats. Like eating this bacon double cheeseburger would be really bad for you. But not all fats are bad for you. Some fats, which are called unsaturated fats, are actually good for you and important to eat and to make a part of your daily diet and intake of nutrients. The emulsification of lipids test, we will fill 2 test tubes with water. Then we're going to add some vegetable oil to each of the test tubes. Notice right away that lipids are non-polar. Water is polar, but lipids are non-polar. And so we say things like oil and water don't mix. In order to get oil and water to mix, we need something called an emulsifier, something that will allow the oil to mix a little with the water. So make some preliminary observations about our 2 test tubes. Now we're going to add some soap or detergent to test tube number 2. Soap or detergent is an emulsifier. So when you wash your pots and pans, you use soap that cuts grease. And the idea is it allows some mixing between the oil. We're going to shake up test tube 1. And we're going to wait for a while and see what happens after we've shaken it up. Now we're going to shake up test tube number 2. The oil, the soap, and the water. Shake them up. And hopefully we'll get to see some emulsifying of that lipid. Now after 10 minutes, we see that the test tube 1 has the lipids in a big glob on the top of the water, whereas test tube 2 has the lipids still on the top, but they're not in a big glob. They're in lots and lots of little bubbles. And that's the effect of the detergent, the emulsifier.
Let's take a look at protein structure. Protein consists of carbon, hydrogen, oxygen, nitrogen, and now sometimes sulfur. Proteins are made up of amino acids. Amino acids have this base structure which almost always stays the same, and then they vary with the elements that are, and the arrangement of those elements that are in that R structure. How can we categorize proteins? One way we can categorize them is by their structure. So for example, if I have a long protein chain like this, that's just the primary structure. If that protein chain is bent around, we can call that a secondary structure. If that protein chain is bent around another protein, like this, or folded in an additional way, we can call that tertiary. If we add more and we fold it in odd 3-dimensional shapes, we can call that quaternary structure. So protein can be categorized based on its structures. It can also be categorized based on its chemistry, what makes up those amino acids that make up that protein. We can categorize it based on its function. Enzymes are very specific types of proteins that have jobs that help chemical reactions go faster or better, more efficiently. Proteins are made up of amino acids. We can categorize amino acids into the groups essential and non-essential. Essential amino acids, you need to eat those. You have to get them from your diet. Your body can't make them. Non-essential amino acids, you can make them. Your body can assemble those and put them together. How do you use proteins? What are they good for? Well, one thing they're good for is the structure of things like hair, nails, and muscle fibers. We do get some energy from proteins if we don't have very many carbohydrates available. Notice it's only 4.1 calories per gram. Proteins make up our antibodies to fight in our immune system. We have protein-based hormones, and cells can recognize each other and recognize what kind of chemicals they're getting or are attaching to them or need to get into the cell. These cell receptors are also made up of proteins. So proteins have a lot of uses in our body. So sometimes carbohydrates are called protein savers because they're a good, cheap source of energy. They don't serve a whole lot of other purposes. Whereas proteins serve lots of other purposes for the same amount of energy. It's more economical for your body, in a sense, to use carbohydrates as energy. Notice the difference in these protein structures. Here we have the hemoglobin folded up like this. Here we have collagen in your skin, a little different type of structure. Notice that it's the structure of the protein is very specific to the job it's able to do, much like a tool. If you have a wrench, it only fits one kind of nut or bolt. It can assemble those nuts and bolts, it can deassemble those nuts and bolts, but it has that very specific purpose, like a tool. Proteins are sort of the tools of your body.
This is what we typically think of when we think of cooking egg white. But that's what happens. It goes from clear to white. Changing the appearance of meat and the flavor is one way that we can denature or change the nature of the proteins in the meat.
How about nucleic acids? Nucleic acid structure. Carbon, hydrogen, oxygen, nitrogen. Now we're adding this new phosphorus and sometimes sulfur. Usually they're in a helical kind of shape, like DNA and RNA are helical. They typically consist of a phosphate group, a carbon-based sugar, and some kind of nitrogen-based combination. What do we use nucleic acids for in our body? Well, each different type of nucleic acid has a little different type of use. So, for example, DNA is going to store your genetic information so that it can direct how your cells put together proteins and use them. RNA is going to be what builds the proteins, protein synthesis, making proteins and putting them together. So, for example, if the DNA codes for it, the RNA will build a certain type of protein that might make your hair curly as opposed to straight. ATP is the chemical energy of the cell. It's like the energy currency of the cell. It's a small energy unit that your body can use for lots of different chemical reactions or lots of different energy requiring or demanding things.